So for this section, I want to go over some other topics, including cloning, site-directed mutagenesis, and really, in order to do that, I want to first cover what I refer to are things in our molecular toolbox. These are the tools that we use and need in order to do things like cloning and recombinant protein expression. Now, when I say the word recombinant protein expression, what that means is we use a DNA that has been obtained from a primary natural source, could be some cells, tissues, things like that, and that DNA is cloned and put into another organism who and that organism then can be forced through a variety of means to produce that protein of interest at great quantities. A great example would be using a bacteria like E. coli to produce a protein. As I mentioned before, I use a protein. I study human liver carbonyl reductase, but we never touch a human liver. We get it all through recombinant protein expression systems. So in order to do any of that, you need a certain set of molecular tools, and I'm going to just kind of go through a few of them. The first one is the plasmid, and I decided rather than try to fumble around with a definition of my own, I decided to use that which comes from the Merriam-Webster dictionary. So a plasmid is an extra chromosomal ring of DNA, especially of bacteria, that replicates autonomously. In other words, it is replicated by the bacteria or whatever it is that carries the plasmid, and it turns out for bacteria, the reason why they happen in nature is bacteria have plasmids that can confer some kind of selective advantage for them in terms of survival. And so if they have a plasmid that contains a gene that can keep them alive, then those bacteria go on, reproduce through binary fission. They make copies of those plasmids, and the bacteria that have the plasmids have an advantage, a survival advantage. And so that's how they've kind of come about. And now we can use them in a laboratory setting to cause bacteria to produce proteins of interest for us. So what you see here is a example of a schematic of a plasmid. This plasmid contains about 4.4 kilobase pairs. So in other words, kilobases. So in other words, it's 4,376 base pairs are part of this. It's double-stranded, and it's circular. And this is an example of a plasmid that was made in a laboratory. It's PBR 322. And in this plasmid, there are several features that are kind of interesting. One is they can confer ampicillin resistance. So bacteria who have ampicillin are exposed to ampicillin and carry this plasmid will be protected because, and the same thing with tetracycline. So what this does is it makes it possible for a lab technician or whatever you want to say to force a bacteria to keep the plasmid, because otherwise the plasmid will take up valuable resources that the bacteria has, and if it can, it will get rid of that plasmid some way, somehow. But if it needs the plasmid to live, then only those that have plasmid will live in an ampicillin-resistant ampicillin, environment or a tetracycline environment. Now, what people can do is you see that there's an origin of replication here. Well, people can also insert other genes into a plasmid, and if they do so in the right way, they can insert a gene, say for human liver carbonyl reductase, and then the bacteria will encode that gene. Will take that gene and make a protein with it if you have the right machinery built in to promote transcription and translation, the making of the protein. So, so, so that's typically what happens. We take the gene of interest that we want to mass produce in an E. coli, and we go ahead and insert it somewhere in the plasmid. Usually there's a polyclonal site where they have a lot of different snipping points where you can snip the plasmid apart and insert a gene into it. The snipping comes about through these things that I'm showing you here: PST1, EcoR1, HINDI3, BAMH1, XMA3, PVU2. These are all examples of things called restriction endonucleases, which I'll tell you about in a minute, which is also part of the molecular toolbox. But their sequences are located in these spots. And so if you go in with the EcoR1 restriction enzyme, it will cut the DNA at this spot. And then you can insert something into the DNA. Say you want to put in a clone gene, you can put it right in the plasmid. And then then the bacteria will be forced to replicate this plasmid over and over and over again, so you can amplify this plasmid so you can make more of it. And as long as you have a reason for the bacteria to keep the plasmid, such as ampicillin resistance or tetracycline resistance, then it will, and you can manipulate then the bacteria to continue to keep the plasmid, make more of it, or eventually you can use a plasmid to help a bacteria make protein. And so that's kind of one of the key things that are really important. So if you know, plasmids then are the circular DNA that contains all of the stuff that we want to force a bacteria to do. If we wanted to make more plasmid, we could do that, or if we want to encode a protein and have it make the protein, we can put it all on a plasmid. We don't have to do anything to its chromosome. A bacteria has one chromosome, and so we don't have to play around with the chromosome. So that makes working with DNA and doing things like inducing protein expression and stuff like that much easier if you could do it with a plasmid than having to manipulate the chromosome. So anyway, that's one of the tools in the molecular toolbox. Really important. Now, here's another tool. This is something I just got done mentioning: restriction endonucleases. And restriction endonucleases are just an enzyme, and endo means that it can cut within a DNA molecule. So it's not an exonuclease. Exonucleases will cut on the ends. Endonucleases make snips inside of the nucleus, of the DNA. So here you see a double-stranded DNA sequence, and you see the thing shaded in green. Whenever this sequence is present, and then you treat it with the restriction endonuclease BAMH1, it will cut right between these two Gs and these two Gs, and it will cause a SNP like that. And there's overhangs that base pair. You see that? So there's a TAG, a TAG. So they actually base pair with each other. So they can come back together, and you can hit it with DNA ligase, which will stitch up the cut parts. Or what you can do is you can take a gene of interest, and that gene just has to have GAT added to the end of it, okay? And then you can snip it right there and get it to fold in, and you can insert a gene by simply cutting out a piece, cutting it with BAMH1, for instance, and then going ahead, and if you have a gene and it has something like this on the end, where there's this would be TAG, okay? Then you would have a gene like this, but there's your base pairing and all that. Then you can insert this whole chunk right in here. So this will base pair with this part will base pair with this part, and you can actually get it to kind of fit in like a puzzle. And you can be very selective and put a gene right into a plasmid by just cutting it with restriction endonuclease. And then you have to allow DNA ligase or something to fix the breakages that would be on either end, on all the ends, actually. All right, so another type of endonuclease, so this would be called sticky ends, is how we would say that. Another way would be to have another type. Here's small one, SMA1. This one will cut when it sees this sequence. You see it's, by the way, you notice these are palindromes. A lot of times you will see that sequences that get cut by restriction endonucleases are in fact palindromic. That is, you can see we got GGG, GGG, CCC, CCC, so they kind of fit that way. You'll see that pretty commonly. In this case, it doesn't cut on staggered ends. It cuts right between there. And that generates what we call blunt ends. They're not sticky. There's no overhangs. And what this allows is you can actually take any gene of any interest you want and just shove it in here, let it kind of insert at DNA ligase, and it will snip it into place. It's non-specific. It'll put any kind of chunk of DNA you have here, including closing this back up again. So the trick is you want to add more of the gene itself so that there's a whole bunch more, so the odds of it falling in place correctly to align, and so that this could outcompete just the plasmid being resealed again. So those are kinds of things. Restriction endonucleases, then you can think of them it can be very specific molecular scissors, DNA scissors that cut at certain areas, and then it can allow you to put in or insert a gene into a certain spot in the DNA, and in particular in a plasmid. That's usually what we think about, and it makes it possible to do cloning and stuff like that. This is kind of a similar. This is EcoR1. I think it was the first ever endonuclease that was identified, and you can see it has its own sequence again, and it's palindromic again. You can see you got CTT, CTT, GAA, GAA. And so when you get a cut, then you create these sticky ends, these overhangs. And again, all you got to do is, if you want to insert a DNA in place, all you got to do is add a piece of DNA that has, the right sort of ends on it. So this would be, I flip-flop these. Let me write it this way. There we go. I just hit it wrong. So then this would be A, T, T. And if your gene has, on this end, has T, T, A, A, it allows you to just plop it right in place. So this will base pair with that. This part will base pair with that. You can insert then whatever the gene is in between, and then hit with DNA ligase. And again, that will help insert the gene in place where it needs to go in order then to be expressed by E. coli or some other bacteria using the plasmid. So now that brings us to a real game changer in the field of genetic engineering, molecular biology, and that is polymerase chain reaction. It has found use in making site-directed mutants, which I'll talk about later. It also is used to quantitate RNA transcripts like messenger RNA, or to create DNA, amplified DNA from an RNA transcript, and that process is known as reverse transcription polymerase chain reaction. and people can create then DNA, what we call cDNA, from the mRNA transcripts. So anyway, the bottom line is, before I get in, I'll talk about those things later. I'm just bringing it up now. In essence, PCR turns out to be like a molecular photocopy machine. You can use it to amplify segments of DNA for a variety of purposes. It becomes a nice way to do site-directed mutagenesis. It becomes a nice way, like for instance, you watch these Forensic Files or some other shows. PCR has really changed the way forensically that DNA can be used to either identify victims or criminals in the committing of a crime or involving the addressing of a crime. So PCR is quite a cool thing that has, like I said, changed the landscape of biochemistry, molecular biology, etc. Now, how does it work? Well, it uses a lot of the same things that are needed for normal DNA replication. DNA replication is making new DNA. So the technique itself harnesses many of the items that are actually used in nature to replicate DNA. So one of the things DNA replication requires is something called a primer. A primer is a small piece of DNA that an enzyme that makes the new DNA is called DNA polymerase. You can see right here. DNA polymerase requires a starting point from which it can add on to the growing synthesized DNA strand. The other thing you need is a template, and the primer matches up to the template. And so you can see here's the template is the original maybe gene that you want to clone or something like that. And so what happens then is you enter in primers that kind of mark the ends of where you want the DNA to be amplified. And there is again, with DNA, you have two strands. You have 5' to 3' end, and what we call the reverse complement of that. So you have 3' to 5. So you have a 5' to 3' and a 3' to 5', and they base pair just like you've seen or learned earlier. And what you do is you use a series of temperature melting. So you increase the temperature, and that causes the parent strands, the template, to separate in its two strands. And you have an excess of the various primers. You need the primer that can match up to the 3' to the 5' strand, and you need a primer that can also grow from this 3' to the 5' strand. And so you have a 5' end and a 5' end here, and those base pair with the template strands. So these primers you design that have just the exact right base pairing capability that target the beginning and end of the gene of interest. And so since you have an increased temperature, it melts and prevents the DNA from coming back together. When DNA comes back together, we call that annealing. How do you spell annealing? I think this is it. So annealing, that's when the DNA comes back together. So so then what you do is you lower the temperature. So this step, you increase the temperature, then you lower the temperature, and you have excess primer, way more primer than you actually have template strands, so that the primer has a competitive advantage to base pair with the targeted regions. So when you lower the temperature, you need to lower it, but not so much that that you really promote lots of annealing. You lower it usually just below the melting temperature, sometimes 60 degrees or whatever Celsius, something like that, 68 degrees Celsius, where here you might be, initially you might be in the 90-degree range, then you drop it down to like 60 degrees, and then you pick a DNA polymerase that can actually work really well at that temperature. Usually they get those from what are known as thermophilic bacteria. Thermophilic bacteria have the capability of living sometimes in like boiling levels of so you're not going to kill the DNA polymerase by heating it up to 90 degrees, because it comes from it's well suited for living in a very, or I shouldn't say living, surviving very hot temperatures. So this DNA polymerase is present, and then there's a bunch of nucleotide triphosphates that the DNA polymerase is going to use to fill in the growing strand. So what you do is you kind of know the speed at which the DNA polymerase can fill in base pairs, and you allow the DNA polymerase to just go and create new strands. So here's a new strand and here's a new strand. Now this is this kind of brownish blood red color is going to be one of the daughter strands that forms, and it has a primer right here. And then this is the other daughter strand with the more bright red primer present. And so then you lower the temperature, and by lowering it, you let the polymerase fill in, do the work of filling in, and then you go ahead and you heat it up again. You put it through another heating cycle. So you increase the temperature again. That will cause things to melt, and then you lower the temperature again to the operational levels. And so when you heat up, you're going to get primers that are forming on the template strands, just like they did originally, okay? But you're also going to get primers forming on the new daughter strands that were made, okay? And so what happens is you start getting daughter strands get copied as well as the original template strands. And so you get, you know, a series of daughter strands that are being replicated, and a series, and so you get granddaughter strands, I guess you could call those, and new daughter strands. Okay, so this happens. Now you, guess what you do again? You heat them up again. So you increase the temperature, decrease the temperature, and what happens is you start seeing the daughter strands themselves reacting. So now all you get are these little chunks of DNA that these are made when daughter strands make new daughter strands or granddaughter strands. I don't know if that's a phrase, but I'm going to use it. And so by continually increasing the temperature, decreasing the temperature, giving time for the polymerase to fill in things, you start now, once you hit this point, you know, you're still making copies of the original template strand, but now you get the product that we're looking for. The nice, tidy piece of DNA that forms when you have the different primers filling in. And now once you hit here, it's binomial expansion. I mean, every cycle is going to double the daughter strands that of interest. So you go through several additional cycles. Maybe you do 16, 17 cycles in all, from the starting point to the end point. And what you get is a big pool of replicated daughter strands that then leave you with huge amounts of DNA now that you can use for further work. And this is where you can use restriction endonucleases or other things to cut and insert and do all kinds of things like that. So this has been a very powerful technique. I've used it many, many, many times. The trick to understanding this and why you get these nice short strands has to do with the fact that if you look, when you start replicating the daughter strands, what's going to happen here, if you look right, say here, all right, you have one short piece, and then you have this new primer right here that makes a new strand in that direction, and pretty soon you're limited by the boundaries set by these original primers that fill in the ends, and so that's what you're seeing here. So, because the DNA starts where the primer is, and so there's no DNA around in either direction to fill in, because the primer, fills in that direction, and etc. Anyway, bottom line is you get a bunch of these fragments of just the desired size and sequence that you need. So that's kind of the general idea. That's how polymerase chain reaction works. And let's continue on with this story. So now that we've gone through the molecular toolbox, restriction endonucleases, PCR, etc., now we can look at how these things can be used to do the things you've heard time and time again, things like cloning, etc. So we got plasmids, we have the PCR, restriction endonucleases, and now this allows for cloning and subcloning and eventually doing some other things. So for instance, in my research laboratory, I mentioned this before, I have DNA clones of human liver alcohol. I also have alcohol dehydrogenase from human liver. Human liver carbonyl reductase clones. And what you do is I have to go yell at my dog. 1 minute. Little squirt just robbed me. Took something out of the garbage. All right, that's what puppies do. Little jerk. So some of the things you can do, for instance, let's say there's some DNA that you want. That's my dog playing with a paper bag. Let me stop him for a moment. Okay, I just put him in his kennel and gave him a treat, so hopefully that will keep him out of my hair for a little bit here. He's 6 months right now. Cute little guy. And annoying. Okay, back to business here. All right, so let's say you have some kind of DNA and a part of it contains some information that you want. Now, there are many different ways that people can clone. Cloning is simply just making a copy of the desired gene or piece of DNA that you want to clone. A lot of times what you do is you'll use PCR to amplify a region of the DNA, and then you'll insert it somehow into the polycloning, multi-cloning piece of the plasmid, which usually what happens there is you'll have a restriction site that you can cut open like this, and then you can take a restricted piece, so maybe your part that you copy can, like me, sometimes what I would do is find a range that includes the gene of my interest, but also includes a restriction site. So sometimes we can actually have a little bit of what we call an overhang in our PCR. So what we would do is, I'll show you what that looks like off on the side. So let's say I want to clone a piece of DNA using PCR so I can amplify it, but I want to make sure that at the end I have the right overhangs so that I can use a restriction cutted plasmid and then have my gene pop into place. Well, there's techniques for doing that, and I'll give you one idea of how this could work. So I'm going to just kind of slide off to the side here. So let's say I have my piece of DNA. I have my double-stranded DNA. I'm just going to use two colors just to kind of keep it straight. So let's say here's a double-stranded DNA, and I create a plasmid. I'll just use a primer that can base pair right here. Actually, I'm going to flip this over Don't write anything yet. I'm just going to erase that part. Okay. Let's see, I create a primer that base pairs over here and then goes that direction. Okay. But my primer, what I can do is make a primer that has the base pairs that I want, plus a piece of DNA that will not really, the primer will include a piece of DNA that doesn't base pair. And instead, in this little part right here is where I can put in a restriction enzyme recognition sequence. Okay, recognition sequence. So let's say I want to make sure it in recognition sequence. And then I could do the same thing on this side. I can make a primer again over here, and I can fill in that direction. And on this primer again, I'll put the same kind of restriction enzyme recognition sequence. So when I do several rounds of PCR, what I'm left with is 2 strands of DNA that also contain my restriction endonuclease cutting site. So then I can cut these, and it will leave me with what I'm interested in. It will leave me, whoops, I wanted to change that for a minute. Once I cut it, it will leave me with a DNA. Change it for a minute. I just have to change my pen color. Sorry about that. It will create a DNA that has what I need to now have these overhangs, these sticky overhangs, that I can then specifically splice in to my plasmid. And so that's essentially what you're seeing here. So you can use PCR to do cloning. It leaves an overhang, and then you can fit it into your expression plasmid, has your ampicillin resistance gene, just like everything I talked about before. And now you have a working plasmid that contains a clone of the gene of interest. And you can then, depending on the type of plasmid you use it to just make more DNA. So sometimes there's something just called a cloning plasmid. It's not used for making protein. It doesn't have the proper machinery to make the cloning, to make the protein, but you just simply transform it into bacteria, and that's what we call the process of putting a plasmid into bacteria. And then you grow it in the presence of we grow it up in the presence of ampicillin, so only the bacteria that keep the ampicillin gene actually gets protected. So the bacteria that don't get the ampicillin gene, they die. And then there's going to be sometimes what happens, and this part is not really well talked about, but there's something called the LAC-Z gene. And the LAC-Z gene contains something called beta-galactosidase. And so there will be some plasmids that do not get the piece of DNA that we're interested in. They just close right back up again. That would be this one. And so when they close right back up again, the LAC-Z gene is not disrupted and beta-galactosidase can be made. Then what happens is, when one of these pieces go in, they'll block the LAC-Z, so they'll disrupt the LAC-Z gene, which encodes for beta-galactosidase. And so plasmids that we're interested in do not have the ability to use lactose because they lack beta-galactosidase, which is used for making lactose. So when that happens, though, and we no longer can make DNA, so once we've got all these transformed, then we grow all the bacteria on a plate in the presence of a series of lactose with some other color things going on. So when the lactose gets used up, it generates, the colony becomes blue. And the only way lactose can be used up is if they have an intact gene of the beta-galactosidase. And so all of the blue colonies represent plasmids that did not get our gene. But the white colonies represents the plasmids that did get our gene. So what people will do then is select these white colonies and then replate them on a plate, and they'll grow a bunch of new colonies. And these colonies contain bacteria that contain our plasmid, and then we can make more plasmid simply by growing the bacteria. That's a typical way to clone something. Sometimes you can then further subclone, so you can use PCR again, and you can put the bacteria into a plasmid that not only is good for doing the cloning and determining which things actually got the recombinant piece of DNA, but you can also use them then to make protein. So to make the protein that's on the gene. So in my lab, for instance, what we did was we took a plasmid called PET5A, that's the name of the plasmid, and we essentially cut out a piece, and we put in my gene of interest for carbonyl reductase, and it now is used to make protein for us. So we get our carbonyl reductase protein. And so this is how we make human liver carbonyl reductase. And we didn't do the initial cloning, but we did do the subcloning, it's called, where we were able to take the cloning vector, the plasmid that contained the gene but couldn't make protein, and then we took that gene out and put it into the PET5A plasmid. And that was one of the first things my lab ever did that was successful, back a long time ago. So it was really cool. I was pretty happy and excited to have this experience cloning something. I just thought that was cool. Okay, so to be clear, to clone, you use PCR, and you have to have the right kind of DNA to do this. Now, if you're using eukaryotic DNA, the problem with a gene in a eukaryotic DNA, I'm going to show you something, is that a gene in eukaryotic DNA, I think I mentioned this earlier, but I'll say it again. I'm just going to use a little marker color here. What happens is the gene contains fragments of DNA that actually can be used to make the gene, to make the protein, but then it also has pieces of DNA in between the fragments that don't code for anything, or at least don't encode for the gene of interest. So when you make the RNA from this, okay, so you have your DNA and it gets turned into RNA, right? What you're going to get is an RNA strand that contains the region of interest, the region that doesn't encode for the protein, the region that does, the region that doesn't, and the region that does again. So you get this kind of arrangement. And this, at this point, we don't really call this part, we call this, we don't call it messenger RNA, we call it heteronuclear RNA. And it contains the parts that encode for the proteins. It also, in these we call exons, because that's what they express. So that's how you think about it. They express the protein, exons. But then there's a part that needs to be actually removed, and those are called introns. And we've already covered things like RNA enzymes. Sometimes RNA enzymes can help remove the introns, and some other introns are removed by a group of proteins that can help snip out the introns. But essentially, without getting into all the details, we wind up with all the exons stitched together to make our messenger RNA. Whoops, wrong button. So here's where the mRNA gets made then. And then the mRNA, and here's the trick when you're cloning in a eukaryotic, because this happens in eukaryotes. The prokaryotes, like bacteria, doesn't really happen that way. The genes are already all exons essentially, and so cloning's much easier in that case. But what people do is they can actually use reverse transcriptase, PCR, so RT-PCR, and they can make DNA from the RNA, and we call this clone DNA. And this is the cDNA, is the intact gene ready to be produced, ready to produce a protein, And so what you can do then is do this for a whole cell. And what you get is something called a cDNA library. So you let the cell go through all of its stuff. You look at all of the mRNA that gets made. You do RT-PCR, and you get a cDNA library. Then that's that cDNA library that you can use and do the cloning procedure that I just covered. So it can be kind of complicated, but it's the techniques that we use. And what's really cool about all of this is we are just harnessing the natural enzymes that it's their job to do what they do. DNA polymerase makes new DNA. Reverse transcriptase comes from viruses that can take a RNA and turn it into DNA and use that. So there's these things just happen in nature, but we take them. We might do some genetic engineering on them to give them some kind of other properties, but we're just using the natural things around that happen in normal life and using them to make pieces of DNA, to clone, to just use those things, which I find really fascinating, kind of cool. So the last thing I'm going to talk about is mutagenesis. And there's lots of reasons to do mutagenesis. You can do mutagenesis. Probably the most common reason why it's used is you want to change an amino acid at a certain position in a protein. That would be one of the things you'd want to do. The other reason might be if let's say you're studying one of those catalytic RNAs and you want to change one of the bases from, say, adenine to a cytosine. Well, you can do that using mutagenesis. And so the techniques, there's so many of them. You can use PCR methods, and you can use some other types of methods. The one that I'm going to show you isn't really a PCR method, but it uses much of the similar technology that you would see in PCR. You would use polymerases, you would use a thermocycler, which would change the temperatures as needed. But what happens in this method is you never actually make copies of the daughter strands. All you do is make copies of the parent strands. So the template strand just gets copied, but you don't copy the copies. And so what makes this a very nice technique, it's called quick change. And it's the technique that I'm going to have you look at in your workshops, or you may have already started on that actually. But this technique uses something, I guess, that you could say looks like PCR in its operation, but it does not copy the daughter strands. So here's what happens. You identify the region, the gene of interest, and the position of interest that you want. So this, that's what this is showing here. Here's, let's say you want to change the site, and you do this in a plasmid. So you start with a cloned gene that's in a plasmid. So I use this technique lots. And so you find you find out where that difference is going to be, let's say you want to change a glutamate to glutamine. And that's just a matter of looking things up on the codon table that I told you about earlier. You use that codon table then to figure out how to change the amino acid position at a given site. So once you figure out where you want to change and what you want to change to, then you create mutagenic primers. So these primers are going to be used in a thermocycling sort of thing that looks a lot like polymerase chain reaction, but as I mentioned before, the only thing that gets copied you're going to see are these parent strands, and the daughter strands don't get copied. And there's a reason for that, and I'll explain in a minute. So you encode for your mutation in the primer, but you have to make sure that there's enough of the primer that can base pair on either side of the mutation, because when you introduce a mutation in your primer, it's not going to be able to base pair with the native position. So your primer is going to include the mutant that you're interested in. So typically, and you'll see in the workshop, you'll design a primer that has a certain length to it, and you're going to want to make sure that it has good matching on either side of where the mutation is to make sure that enough base pairing happens so that you can get the copy that you need. Then you go through the thermal cycling. That's where the temperature cycling comes in. So you heat things up, the plasmid melts, then you cool things back down again, and that allows your primers, which are added in excess. And so what happens is the primer, and we use DNA polymerase here, and the primer fills in all the way around the plasmid until it meets up with the primer again. And you get it in the opposite direction too. Now, when these plasmids are, when the daughter strands come together, you get a series of plasmids like this. But I want you to notice that there's a nick in the DNA. And so that's why you can't fill in. Let's say you had a primer that matched here and it would go around this way, right? But it can't because there's a nick right there and it prevents that strand from being copied. And the same thing is true going in that direction. So what happens is you have to ignore that. You won't get copies of your daughter strands because of that. So what you're going to be left with is a few parent strands, okay? A few parent strands, and a whole lot of daughter strands because you keep copying these templates. And so this is much more like a true photocopy machine. A photocopy machine, if you think about it, you put in your originals and you just make copies of the originals. You don't use the copies of the originals to make new copies. So PCR, you make copies of the originals and copies of the copies, if that makes sense. But in this technique, you're only making copies of the originals that also happen to have this primer that contains the mutant. Now, here's something that's kind of interesting, and the reason for not making copies of the copies is because you want to make sure that you can then, in the end, get rid of your original copy, because it contains the native wild type sort of arrangement. So if you're, let's say, using my example of earlier where I said, let's say you want to take a glutamate and change it to a glutamine. Well, this one's coded for glutamate, whereas your daughter strands are encoded for glutamine. So you want to get rid of these parent strands. And what's interesting about them is, since you're using a plasmid that you've purified from E. coli, is they have methyl groups attached to them. So you have methylated DNA. And that's one way that bacteria keep track of the methyl of parent strands and stuff, is through this methylation. This is something called epigenetics. So anyway, so you get the methylated parent strands, but these strands, since these were made in a test tube and not in a bacteria, they don't get methylated. So there's a restriction endonuclease called DPN1 that just cuts methylated DNA. And so by cutting up the methylated DNA, you're getting rid of your parent strands, but you're keeping your daughter strands. Now, there's a big problem here. These are actually not fully stitched up plasmids. These things are actually needing to be repaired. So the way that you do that is, because these things can actually base pair and look like a plasmid, you can transform these into bacteria, and the bacteria contain DNA ligase that can fix up the nicks and lead to actual real plasmids, and then you can let the DNA, the bacteria, make more plasmids, and then you can use these plasmids to make protein that has now the difference in position. So maybe, again, we said, let's say we wanted to do a glutamate to glutamine. Well, these guys will put glutamine where the glutamate used to be. So in a nutshell, that is this technique, and that's the technique I'm going to teach you to do. And you may have already worked on it at this point. There you have it. So that's all I want to talk about with working with DNA. There are methods for sequencing DNA and stuff like that that I could get into, and you can read about that. But the truth is, these days, I'm going to skip that. If you want to learn more about that, you can look it up. There's plenty of stuff on DNA sequencing. Okay.